quarantined like 12 days ago because of a class.
Awesome. So it is my pleasure to introduce Dr. Wenfei Han, who is an assistant professor in the Department of Neuroscience at the Icahn School of Medicine at Mount Sinai. Dr. Han received her doctoral degree at Tongji University, where she gained both basic and clinical training in orofacial and gastric surgery.
Yeah.
She then pursued postdoctoral training at the John B. Pierce Laboratory at Yale University in the lab of Dr. Yvonne Diarujo.
Yeah.
And Dr. Han's work combines complicated central and peripheral surgeries with modern neuroscience tools, and her recent findings have really applied these techniques to study a novel role for vagal gut-brain signaling in food reward. And I think we'll hear about some of that today. And so Dr. Han, thank you so much for coming and speaking to us here at Monell and beyond. We're really looking forward to hearing about your exciting research.
Okay, thank you. So now I will start to share my screen. And okay, start. Okay, now you can see my screen, right?
Yeah, that looks great. We can see your slides.
Okay. So we can, and you just don't see me, but that's okay.
Looks
okay.
Good.
Yeah, okay. So hi, today I'm going to talk about vagus nerve and the physiology of reward and digestion. So here's the animal with neuroepithelium and the tongue, right, with the taste bud. So over the last century, like there's always a well-accepted model of how food intake works. So we basically believe in a textbook knowledge that there's a system located in the oral cavity that transmits the hedonic quality of the food to the brain, plus flavorable flavors. So this cranial nerve that innervates the tongue and the neuroepithelium send information to the brainstem that drive the food intake reward and the motivation, and so the classic thinking of this is the motivation part to drive the food intake. However, there's another. So there's always like a counter position. People think of gut as a negative feedback, like okay, we have enough food in the stomach, so we are already bloating, so it's time for us to stop eating. So there's always this model that the oral drive the hedonic value and the gut drive the negative feedback, which working together control the food intake. And then for the gut information for the gastrointestinal tract, so one of the cranial nerve, the vagus nerve, played the important role as I'm showing here. And so the vagus nerve, it's one of the largest cranial nerve. As it leaves the skull, it goes to innervate most of the visceral organs, especially the gastrointestinal tract. The main function described to the vagus nerve is as a key player of negative feedback of the body system. So the innervation of the gut, especially the stomach, upon bloating and filling up the stomach, send a negative signal to the brain and tell us to stop eating. And this dual system has been considered basically the way the feeding system works in most animals because the vagus nerve is highly conserved. This is believed to be true for every animal. However, the problem really is that when you look at the data more carefully, and if you look all the experiments where no matter human or animal, when their vagus nerve was damaged or being cut, you just don't say this animal or human eating more, it seems like there's not this negative feedback system. So because this data doesn't really fit this negative feedback model, and so we want to know like what the details, like what the vagos really tell about what's the real function of the vagos in the food intake. So our lab have working on gut-induced reward for years. So we developed a preparation of injecting nutrient directly into the gut and the measure dopamine release in the striatum simultaneously. So with capsaicin and the vagotomy approaches, we know that the vagal sensory fiber is important for the midbrain dopamine system to detecting lipid nutrition in the gut and send information to the dorsal striatum. And back then, we knew that the right nodose is sufficient to maintain the normal response. However, the complicated anatomy of vagus nerve made it very difficult to manipulate the study in a clean way. So here I'm showing you a mouse. The vagus nerve innervates many organs. One important thing to keep in mind is what we call the vagus nerve is actually a set of fibers composed two completely separated group of neurons, right? So in one hand, we have a group of sensory nerve locating outside the brain in the periphery. And so it's this one, right? And on the other hand, so this periphery sensory neuron, we call the nodose ganglia in small animal, and in a larger animal like a rat or human, it's split into nodose and jugular ganglia, and where the nodose ganglia sit the sensory neurons that innervate the gut, where the jugular have the neurons innervating the lung and the heart. And this sensory fiber are monitoring the state of the visceral organ and then send information to the lower brainstem, a lower brainstem called the nucleus of solitary tract. And so from there, the information travels upward. And in addition to this, the second group, which is the green, which is the motor neuron, that it's the parasympathetic vagus neurons. The cell body of these neurons sit in the brainstem, and these neurons send their fibers through the vagus and reach to the organ. So these neurons passing through the nodose ganglia. So given this arrangement, you can appreciate that whenever you try to cut or stimulate the nerve, you are going to almost inevitably stimulate or damage both the sensory and the motor function, both part motor and the sensory function of the vagus nerve. And also because the vagus nerve innervates more than one organ, so that's why the literature is rather confusing, because it's difficult to manipulate this set of neuron in an organ specific and in a sensory versus motor specific way. So to solve the problem, we apply an approach that I developed in my earlier study. And so what I was doing, what I'm showing here with this cartoon, so we inject a retrograde virus, carry the cre recombinase into the upper gut, which is the set of interesting for us. So the cre is going retrograde, travel to the nodose ganglia, and also to the parasympathetic motor neurons, because they both innervate the same part of the gut. Then we perform the second injection into the nodose ganglia of a cre-dependent channelrhodopsin. By doing this, I can target specifically the sensory part of the vagus link to the upper gut in a left or right manner. So we can stimulate only one part of the, like only the sensory part of the vagus neuron and leave the parasympathetic part of the vagus nerve untouched. So when we say the first part, so it's another drawing of the virus strategy. So when I inject retrocrete in the upper gut and create dependent channel rhodopsin in the right nodules, here we are visualizing the terminal of the lower brainstem. So this herring-like structure arising from right nodules carry the information from upper gut in a weight mailing the NTS, the part, and we don't see much of the air postrema like being innervated, and the air postrema is much more linked with aversion. And then on the other hand, when I did the same injection into the right nodules, and so this neuron fiber innervate in a very different area. So we can see here very densely innervate the air postrema. And so we can see here at this level, there's this very obvious asymmetry. And but one important point that I want to raise here is this: the asymmetry depends on which part of gut you are targeting. And so here, when I target the retrocrete in the lower gut, including the ileum, we see a much greater innervation of the area postrema for both left and right nodules. The asymmetry between them is less obvious. That's probably why undigested food in the lower gut is not a signal of reward, but quite the opposite, which can cause aversion and nausea. And also for the practical point of view, this reveals the limitation of using a molecular marker in the nodose ganglia that presumably innervating the whole gut. And because this marker do not capture the subtle distance differences between the upper and the lower gut. And finally, I implant an optic fiber over the brainstem and just over the terminal of neither left or right nodose fiber. And so this approach allowed me to stimulate the sensory terminal that convey the information from gut to the brain. And the other advantage of this approach, it's of course I won't stimulate any vagal innervating to the heart or lung, right? So this part was untouched. Also, the motor neuron of the vagus nerve I was not supposed to touch. So we start with a very simple behavior. So in this behavior setup, the animal in the behavior cage, when the animal poke one side of the hole, he gets a pulse of a laser, and the other side, he don't get anything. So if the vagus, it's a negative feedback information that convey the fullness or bloating information to the brain, the animal would like just leave it alone, like never poke it again. However, when we do the stimulation to the right nodose terminal, so this stimulation actually very attractive to the animal. So the animal would come back and looking for stimulation by himself. So this repeated like the animal searching for the stimulation by himself. In the behavioral neuroscience, it was called self-stimulation. It means the animal evaluates the sensation they obtained from the vagal stimuli as attractive. So this is very interesting and also completely surprising if you think the negative feedback serial of the vagus nerve. So the behavior was very similar to what was described in the early 50s and 60s as a self-stimulation behavior, which was guided by stimulation of what was called the pleasure center in the brain. James Auld was a physiologist working on brain stimulation in rats. In one particular rat, James missed the target and he realized that rat would go back and approach the stimulation again. He discovered that the animal would compulsively self-stimulate the brain region again and again, even give up food or sex just for the stimulation. So at that time, this was a big revolution in the psychiatry because they were believed that only an active state like hunger or thirst would drive reward behavior, but actually he found the central neurons, like certain neurons in the brain, are sufficient to drive the self-stimulation in the absence of any physiology drive. Yeah, so sorry, my previous drive. And so this, and so what I want to also using another behavior test. So I have this animal in the cage, right? So we can see here one side is the animal leg, so it's a softening, relaxing bedding, and the other side is this flattening platform the animal usually hits. So if I pair the laser only when the animal stay in the flattened part, and we can see how the animal will behave. And so you can see the mouse without laser is just hanging around. He like more the bedding side, and he don't like the flattening surface. But yeah, he like the softer part. We wait a little bit. Yeah, but when I turn on the laser, when he's in the side that he shouldn't like, you can see he actually like hesitate, right? He's like, okay, maybe I should go to the bedding, but I think I like the smooth part more. So you can see clearly the animal switched their preference just because there's laser in the side that they're supposed to not like it. So it means that there's some immediately information coming from the gastrointestinal tract that drives a preferred information in the awake free behavior animal. And here is the codification of the data. And this work only on the right dose. And here we also did a flavor nutrient condition, which is just we pair a non-caloric flavor with the vagus stimulation. And the animal also will show very clear preference for the flavor which was paired with the vagus nerve. So the reason why the vagus nerve seem to produce this type of reward-related function is because it seems to be somewhat connected to the brain reward system. So what are the brain reward center, right? So one of the critical region in the brain coding the motivation is the so-called nigral, the substantial nigral that produce and release dopamine to the dorsal striatum. And so from the previous study, we know that releasing dopamine from nigral to dorsal striatum is fundamental for all the motivated behavior. So if you destroy these neurons, the animal will no longer acquire any motivated behavior. They lost interest for food or sex. So in agreement with this, we observe that when we stimulate the sensory innervating neurons from the retinal dose, we observe a significant increase of dopamine release to the dorsal striatum. And this is very similar. If you remember, I show you the slides that when I infuse a lipid directly into the stomach, the dopamine also increased in the dorsal striatum. So however, when we did the same preparation onto the left nodules in the waiting fibers, the dopamine level didn't change. And we also observed that around 20% of the dorsal striatum neurons responded with increased firing rate to the stimulation of the right nodules. So one interesting question for the future is whether this neuron that filter the vagal signal from the gut comply with the classical computations associated with like a dopamine system, like a reward prediction error and so on. So now there's a question. It's like how this information from gut go to the negral heart. So we need to mapping the pathway, and so we know that this lower brainstem is so far away from the midbrain dopamine system. So how the information reach there, and so we use the same strategy that we combine a retrograde injection in the gut, and then also a crate-dependent anterograde polysynaptic virus into the nodose ganglia, and then we can see how the information from the gut spread to the brain. So here is the sensory fiber in the gut, and then the nodose neurons in the right side. And here we see the HSV in the aeropostrema and the solitary tract, very similar to the channel rhodopsin fiber innervation. And then we see after the NTS, the cells jump, the virus jump anterograde to the parabranchial. And what is interesting is that this information, they avoid the part of the parabranchial that was known to be aversive, which is the external lateral. So it's interesting, right? So the information was separated at like a relative early phase. Here is the pons, right? And then from the PBN are glutamatergic. So these glutamatergic neurons in the PBN also goes forward to the negral, and also some cells in the ventral tegmental area. And then eventually this information reach to the dorsal striatum. Okay, so here I want to summarize. So why we think right nodules vagal neuron are also reward neurons. So first, the activation can trigger sustained self-stimulation behavior. And the second, as we previously saw, they condition their preference, like positions or flavors, or like to pair the laser. They like the laser stimulation, and they can pair the laser stimulation to a certain condition. And the third, it can induce a self-stimulation neuron transmitter, which is the dopamine, to be released in the striatum. So in summary, vagus neurons are very similar in function to the neurons in the vertebrate brain, including human, and that are known to mediate reward behavior. So we propose that the neuron reward pathway extend to the periphery, including the upper gut in the waiting neurons. The function of this neuron, which is to send nutrient from gut. So for the animal, this is the ultimate reward signal for them to survive. And in fact, a more recent study indicates in human, the vagal stimulation related with reward system. So this is a universal system that conserved across the species. So here you can see the negral has, so this is Waitier, and here we can see the negral also being stimulated with, I forgot to show this. So this is the vagal trunk stimulation. And so actually, we believe this finding may account for a positive effect of the vagal stimulation in the depressed patient. So the stimulation of the vagus nerve is one of the only FDA-approved therapy for the drug-resistant depression. So the patient in a more serious stage with like a suicidal thought that they don't handle well medication, and they can be prescribed a stimulator implanted on the trunk of the vagus nerve. And in clinical trials, it has been proved to work in the majority of the patient. It has been years because a standard therapy for drug resistance patients. So actually this was discovered kind of like randomly. So they examined like a mood rate in the patient who has like epilepsy, and they report like happiness, like in good mood. And so it's important alternatives like in the depression treatment. And a very similar result also observed with just like articular vagal stimulation. Another study showed like a vagal stimulation just from the ear, right? In human, they can boost the drive to working for reward in an operant test. So there's and interesting, there are also some effect of the asymmetry as I observed in animal. And so there's a more generalized effort triggered by the right vagal stimulation, which was not just limited for food. And in human, the dopamine level in response to food seem to have two components. One is the immediately oral sensory input, and another is the delayed post-ingestive response in the dorsal striatum. Here in human, it's pitamal. And so when the patient report they have like a more satisfied, more satiation and less desire for food, and the dopamine level in their dorsal striatum also getting higher. Okay, so now we move to the second part of my talk. So I was talking about the sensory aspect of the vagus. So now I want to talk about the parasympathetic aspect of the vagus. So I did the same type of surgical preparation. I inject retrograde in the gut, and I inject this time the channel rhodopsin directly into the brainstem, the parasympathetic neurons. And then I implant the optic fiber just directly there. And here we show the expression of the channel rhodopsin in the dorsal motor vagal complex. And we can say the expression is restricted in the motor cells of the vagus, and nothing can be observed in the NTS or aeropostrema. And so as we expected, when we perform the optical stimulation, we see a lot of gut contraction using electromyogram, but nothing caused like dopamine release in the dorsal striatum or any reward behavior. So it means that the positive reward part of the vagus, it really only linked precisely to the sensory part of the vagus. And so in order to study the parasympathetic system, so we decided to apply the classic physiology experiment called the Pavlov reflex. So the general principle is that the brain associates a cue with the activity of a gastrointestinal tract. So in particular, the stomach, which produce acidic juice, help with digestion, and all other secretion take place as like to get prepared in response to the food delivery. So to study in detail of the neuron circuit mediating the stomach secreting acidic juice upon food anticipation, we use a mouse cre line which express cre in the ATP4B expression cells. ATP4B was it's a very specific expressed in the stomach pyramidal cells, which function for secreting the acidic juice. So when we cross the ATP4B with the Cre reporter AI9, we can see here that the red cells label the stomach pyramidal mucosa. So these Cre cells will be our enter point for us to understand the brain circuit control the stomach acidic juice release. And so we use a string of it's okay. It's a little disproportion. So we use a string of inducible retrograde and transsynaptic pseudorebivirus with GFP fluorescent. And when we infect the virus in the starting location, in this case is the ATP4B cells, the Cre expression cell will allow the PRV to travel retrogradely, and where the non-Cre expression cell won't permit the virus to jump. So this is a useful way for us to trace transsynaptic pathway in the retrograde manner. And so here when we inject this Cre-dependent PRV in the stomach pyramidal cells, and then we saw very strong label in the cells infected mainly in the dorsal motor vagos, dorsal motor nuclei of the vagos. And so this makes sense because we want to study the motor part of the vagus nerve. So the parasympathetic motor neuron of the vagus clearly involved in this process. And also there may be some NTS, but there's almost nothing in the air postrema, which makes sense, right? To get the stomach ready for the food while we need aversive information from air postrema. And in addition to the vagus complex, we also observe a parasympathetic center. So it's the paracellular, paraventricular nucleus of the hypothalamus. And interesting, we saw a very focused, dense cluster of PRV in the medial part of the central amygdala, which is a more anterior part of the central amygdala, which sending projection down to the lower brainstem. And instead of parental, we also inject PRV into the stomach cholinergic mesenteric cells on the stomach wall that are responsible for the stomach contraction. And we equally saw strong labeling of the dorsal motor nuclei of the vagus, as well as the upstream central medial amygdala. And this implies that this part of amygdala control the vagal nerve responses, not only for gastric secretion, but also for gastric movement. And interesting, we noticed that in addition to gastric secretion, central amygdala seemed to also be very strong labeled with the retrograde virus injected in the salivary gland, both the parotid and the submandibular gland. And so when we inject PRV, so in the end, we think the whole physiology response of the anticipation for the food, including salivation, gastric secretion, gastric movement, all seems to be orchestrated by the central medial amygdala. So this implies that the central medial amygdala is a critical center in the brain driving the Pavlovian response in the digestive tract. And also when we inject the HSV from the gut innervation nodose neuron, we also see a central amygdala being labeled. So we expect that central medial amygdala are not only related with reward, but rather get the animal to be prepared for the upcoming delivery of food. And so when we condition the animal to a certain environment for the upcoming of food, we see a elevated level of safe force expression in the central medial amygdala. So we are very interested in understanding the physiology function of these neurons. So we created a mouse stomach fistula model that we can open and extract the stomach content whenever we need. We develop a contextual conditioning protocol that the animal will present in a certain environment signified with food, and the control has nothing. And so after 3 consecutive days of training on the test day, we opened the fistula and checked the stomach content in the absence of food delivery as we would expect. We observed that the animal were conditioned with food delivery. Their gastric juice is much more acid than the unconditioned animal. And in conclusion, mouse, similar to the dogs, learn to predict the availability of food, and their digestive tract, in particular, the secretory gland of the stomach, are part of this process for the food anticipation. And so when we activate this part of amygdala, that we want to say what we saw the animal display a motor program that is very similar to the type of the program that the rodent display when eating. So they consume, they have this body posture that and also move the falling rhythmically. So, and also you can see they move the jaw in a way suggests that central amygdala produce an overall effect on the animal that signal for the availability of food. And this happened even the food was present. He displayed this type of behavior as if the food has been consumed. So the central amygdala seemed to be involved in a very general system that provide motivation to drive for the animal to eat. Yeah, it continues as long as the laser is on. Okay, so, and also this, also we can say that when I put the EMG wire in the stomach and I turn on the laser in the central amygdala, we can see not only the animal moving the jaw, there's also very strong stomach muscle contraction with the laser stimulation. So this implies that the central amygdala really has the ability to drive the stomach activity. This is a typical response of an animal expecting for food. I think for me, the same, right? When I'm hungry, I feel my stomach is making noise. So this is, so we have been mapping the neuron anatomy of this part of amygdala and the descending projection. We found that amygdala is giving several descending projections that control different group of craniofacial nerves. Central amygdala controls premotor neurons that in turn drive the motor neurons of the trigeminal nucleus that control the movement of the masseter and the digastric. And so the amygdala send GABAergic information to the paracellular reticular formation, which also GABA neuron. By the GABA disinhibition, this pathway, so the central amygdala can trigger the trigeminal motor activation, like working together as closing the jaw via the masseter and also open the jaw via the digastric muscle. And at the same time, the central amygdala provide a very dense innervation through the salivary nucleus in the brainstem, which is also cause the release of the saliva. And finally, amygdala release a very strong influence on the premotor neuron that drive the stomach innervating neuron of the vagus. And altogether, these neurons seem to orchestrate a number of physiology responses that would occur simultaneously when the animal is eating. And thank you for my supervisor, Yuan, and also all my colleagues. Okay, that's it. Thank you.
Awesome. Thank you so much, Wenfei, for a really beautiful talk on your exciting new data connecting all of the, vagal afferents and efferents and all of these simultaneous processes that happen when we're eating. So we can open up for questions now. Please feel free to type questions in the chat, or you can DM me that you have a question and want to come on, or simply just unmute your microphone and hop on into the discussion. And I think I will get us started with a few questions. So my first question, I want to go back to kind of the first part of your talk, where you were looking at stimulating vagal afferents and reward behavior. Yeah. And I was really struck by the video that you showed on the conditioned place preference. And so your data are very compelling that stimulating at least the right vagal afferents can condition reward behavior. But in the video, it happens almost instantaneously. So I was wondering if, does that happen the first time you start stimulating? Does it require conditioning? Or is it kind of like, as soon as they get the stimulation, they know something's feeling good?
Yeah, so for the nose poke, I have more details in my paper. So the animal on the first day, they already show the preference when they post the nose to the hole and they get later they feel there's something good. So and then if you continue train the animal for, I think I did Louis did 3 days, and the animal showed a very clear preference. They learn to go to that hole and get the laser stimulation. And for the place preference, because this is because I run the video 4 times faster. So this is a stimulation for, I think, 20 minutes. So this 20 minutes is enough for the animal to figure out. So you can see at the beginning, the animal was confusing. He started hanging around at the border and he feel like, okay, it's good to get the laser, but the other side, it's maybe also good. And then later he made his decision and then just stayed to get more laser.
So it happens within the first session?
Yeah, the first session within I think 20 minutes. Yeah.
Wow, that's really cool.
Yeah. And we also, when we do the microdynamics, so we just turn on the laser for 6 minutes and we see the surge of dopamine in the dorsal striatum. It's just like no training. You just get there and the dopamine we can see.
Awesome, thank you. So the next question is also related to the first part of your talk, and it's from Mike Tordoff, and he's asking if the site of reward is the gut or the liver.
Oh yeah, because I only inject the retrocrete in the stomach, right? So I don't know what happened in the liver, but interesting, when I didn't show the slides, I was worried about time, but when I did the sugar preference, so in the vagotomized animal, the animal can learn the sugar reward very, very well. So in this case, I inject the very tiny amount of glucose directly into the portal vein, and I would observe a very strong, unbelievable, 50% of dopamine increase in the dorsal striatum. So I think at least, of course, the vagus nerve is for rewarding, but I'm not saying it's for all the micronutrient. I think for sugar, there's more like a pyrolide system guiding the sugar signal. So I think for the sugar, the portal system in the liver is very, very important.
Awesome, thanks. Next is Nick and then Kevin.
Hi, Wenfei. Thanks for the compelling talk. That was really interesting and a lot of data.
Thank you.
I had a couple of questions about the premotor system that you showed at the end that generated the chewing, basically a central pattern generator for chewing.
Yes. Yeah.
So the videos are amazing, but the first question I had was really kind of a simple one. Why doesn't the animal just pick up some food and start eating it?
Yeah, so that's the thing. This video I showed, the animal already had food. They are not hungry. And so when I was working on my previous study, I measured I can recall the very different behavior when the animal is hungry versus is satiated. So when the animal is hungry, so I suppose there's not much information from vagus or from lateral hypothalamus to the central amygdala. So the amygdala, the one that's turn on the amygdala neuron, the animal will pursue the run away and then bite whatever in front of them. But on the other way, if the animal is satiated, he will do this rhythmic movement of the move, like the consummatory. So it's like already programmed there. But how to use this program, the motor system, depends on the innate state of the animal, if they are hungry or if they want to fight. So I think amygdala was like one of the promoter system to control the jaw and the craniofacial system. Yeah.
That's interesting, but you don't ever see that behavior innately, right? An animal doesn't start chewing or pretending it's eating without eating. So presumably there's a gate in the brain, and you were talking about the lateral hypothalamus gating that normally.
Yeah.
So do you think it's state dependent integration and a different region of the brain?
Yeah, definitely it's state dependent. So actually that part data I didn't publish. So Yuan asked me to put a female in the cage, in the hungry animal, like a male, right? And turn on laser, see what happened. The male just go there to lick the female. He didn't bite at all. Yeah, so it's a very like it's a motor system, right? The animal has to do something in the job, but not necessarily biting or like chewing.
So that might explain why I chew my pens.
Yeah.
Thanks.
Kevin.
Yeah, Wenfei, this is very exciting work. Thank you for the presentation.
Thank you.
I think my question is similar to Mike's about sort of these ascending reward signals through the sensory fibers. And I guess my impression of the sensory fibers rising up through the nose is that it's a very heterogeneous fibers. Like some are chemosensitive and some are mechanosensitive, rising from sort of a variety of different levels of the gut. Do you think that a wide variety of these different sort of functionally distinct neurons all contribute sort of to these reward effects? Or do you think there's sort of one specific functional class of cell that is producing this really strong signal that's kind of overriding sort of the sort of negative feedback signals you get from the gut?
Yeah, I don't know, right? Because if you are like saying the COVID, right? You need oxygen, and then there's some oxygen. Then it's rewarding. There's the motivation value of, but it's just right now we are all healthy. We don't have this motivation. But this what I did is I didn't do much. But when I do like inject the retrocrate in the different part, like in the thyroid, and also like in the lung and in the heart, I say there's like a mapping in the NTS. So like if you put virus in the lung and the other part of NTS is dark, and then when you feel the virus in the heart, and the part that was dark was filled with fiber from heart. So there's definitely a mapping, like a topographic, and also it follow the anterior caudal. So the lower gut in the inner way, the relative caudal medial part of NTS, and the lung, and also the neck part in the way to the more anterior, and also like the taste, right? The taste information goes very anterior and the lateral part of the NTS. And the taste is also an example. It could be sweet or bitter. Yeah, based on the property of information, but definitely there's part of the sweet information go to the VTA and then ventral striatum, the animal happy with it. Yeah, thank you.
Yeah, interesting. Thank you.
We have a question in the chat from Alexander Bakhmanov. Substantia nigra dopaminergic neurons are also involved in motor function and are deficient in patients with Parkinson's disease. Can vagus stimulation be used for treating Parkinson's patients?
Yeah, that's interesting. I think actually there's a small paper coming out and then talking about the microbiome and also the gut and information of the yeah, I think the information from the gut is also there are recent paper talking about. I think maybe this vagal information from the gut that in the end adapt in the Negro is related to the Parkinsonian, like the pathology of alpha-synuclein. Yeah.
So are these, so just a follow up to that really quickly, are these the same neurons that are involved in motor function, or do you think there's different populations of neurons within the substantia nigra and a subpopulation of those are receiving signals from vagal afferent signals to stimulate this food reward?
Yeah, actually, Negro, if you look anatomy, right, it's a cover big part. There's a more like a caudal lateral part and a more medial part that close to ventral tegmental area. I think the region that I saw with vagals is a relative medial, close to the ventral tegmental, and yeah. So I don't know, like also I don't think. There's a clear, very good Parkinsonian mouse model so far. Yeah, I think there's more things we can study.
Awesome, thanks. Another question from the chat from Karina Simeon. Thank you for the nice talk. You've said that taste is connected to the vagus nerve. Could you please give more details if you know if there are differences in the pathway for different tastes, like bitter taste, for example?
Yes, so Zucker has many good study about the taste, about the high the wiring of the different taste component. And so I think for the vagus nerve, yeah, I think there's something that many nutritive signaling that reach to the NTS well, like something that you feel nauseous, that some of the fiber was detected by the nodose ganglia, or even like directly detected in the aeropostrema, where there's like more losing blood vessels that they can sense the aversive stimulation directly from the gut. So I don't know how much in at least I think in the upper gut. Oh, I never see the animal like feel like have an aversive effect behavior to the vagal stimulation. I think the aversion concept of the vagals is mainly coming from the aeropostrema that detecting directly the aversive component in the gut.
Awesome, thanks. I had a question about the second part of your talk on the descending modulation of the stomach. And so you did the polysynaptic retrograde tracing, right, from the cells in the stomach, and you showed images from the PVH and the amygdala. And I was wondering if through some kind of time course or follow-up studies, what the circuit is. I might have missed this, but are they all connected, or are they both projecting to the dorsal motor nucleus of the vagus and then downward?
Yes, so the PVH is known to be the promoter of the autonomic system. And I think when I did the anterior grid tracing, so the NTS project directly to PVH. This projection, you don't need to go through the parabranchial. However, if you the information from gut to reach to the amygdala, it goes through the parabranchial. So I think there's a parallel system from the gut. I mean the gut and also the PVH, the other the sympathetic system. They are all parallel, and of course they're talking to each other. And I think the PVH project directly to Niger. yeah, that part is also clear.
Awesome. So lots of interplay between the two, but you think there's kind of these two not opposing, but reciprocal pathways from
Yes,
stomach to the brain. Awesome.
yeah, yeah.
Do we have any other questions? Well, if not, I will end by thanking you for a really fantastic seminar. Arash, did you have a question?
Yes.
Okay, awesome. We'll do one more.
So, Wenfei, amazing talk. I just have a question regarding the distal part of the gut, where you were saying that when you did the retrograde tracing from lower gut, like ileum, and it's always going to area postrema, which is more aversion related. My thinking is like, and when we look for macronutrient digestion, that digestion happens in like different segments of the gut. So some macronutrients would get like digested in the upper versus some that are more like fibers would get digested in the lower. So do you think that that digestion is always going to be aversive because those neurons always project to those areas?
Yeah, so I think for the animal, right, you want your body to work efficiently. So if there's undigested food goes to the lower gut, you should like at least slow your eating. So I would say there's a negative feedback from the lower gut. So it means, okay, you eat too much, you eat too fast. The food haven't been well digested. And I think it's the like at least for protein and the sugar and the fat, mainly absorption is in the jejunal and some lower in the ileum. And then for like maybe other like vitamins and salt and some sugar can be absorbed in the lower gut. But I think to for the, and also if you say the thickness of the epithelium, so the jejunal has this very thick and like big surface areas for to absorb the nutrition. So I think to get the nutrition, like valuable nutrition, it's mainly in the upper gut. That's why there's this reward system linked to the upper gut rather the lower gut.
Just to follow up on that, because there are a lot more studies coming up in psychiatry with role of microbiota in like kind of all these different diseases. So that's all the distal gut. So how are we going to be pushing that the signals from the microbiome, which are like going to be sending signals from ileum or colon, which is even further down?
Yeah, I think at least for the I remember if you say there's this FMR, so the post-digestive signal for each meal is like 20 minutes. So when you have some hedonic things in the mouth and then you wait a little bit, your jejunal start working, and then there's another pulse of dopamine reach to the brain. But when you talk to the microbiome, it's like a more generalized nutrition status. I don't know. I think it's definitely because when I stimulate the left nodules, the animal is not aversive. It's not to try to avoid. It's just not reward driving.
Just to quickly follow up on that before we close, what about satiation signals or peptides released from the GI tract? So, you know, from the proximal to the distal small intestine, there's going to be a range of different peptides released. Are those playing into this rewarding effect that you see on the vagus?
I don't think it's a fast reward, but definitely if the animal is all human, if you lack of one particular nutrition, you definitely don't feel good. But that is more generalized feeling, maybe not this very fast like dopamine in the night. This a fast big wave of dopamine, but more generalized feeling. So like we used to have animal that lack a certain amino acid, the animal just feel really bad. even they have other nutritions, right? But if one important component missing, the animal don't feel happy. Yeah.
Awesome. Thank you so much again for a really fantastic talk.
Thank you.